The goal of this procedure is to demonstrate the steps of creating 3D printed, fully colored, patient-specific anatomical models using data from different imaging modalities. By using the example of chondrosarcomas of the petrous apex, the whole process of image fusion and segmentation, generation of a virtual 3D model, and fabrication of the 3D print are demonstrated. Moreover, volumetric coloring of 3D prints suitable for surgical simulation is described. A variety of imaging modalities, such as computed tomography and magnetic resonance imaging, show different aspects of a surgical site, such as bone, soft tissue, tumor, and vessels. 3D printing technologies offer the unique possibility to combine these different aspects in a single compact and tangible object in real-size dimensions that can be studied and used to simulate the surgical approach. Especially for surgical simulation, it is important to produce 3D prints not only colored on their surface, but providing in-depth volumetric coloring to clearly visualize structures nested inside each other, such as a blood vessel crossing a tumor. This video offers a step-by-step guide for the fabrication of a fully colored anatomical model based on the example of a chondrosarcoma of the petrous apex. The crucial steps in this procedure are fusion and segmentation of medical imaging data, followed by creation of a virtual 3D surface model. As a third step, the virtual model is prepared for multicolor 3D printing, including a modified workflow to allow volumetric coloring of specific parts. Finally, printing and post-processing are described. It is important to use imaging data with high spatial resolution. For example, a slice thickness of 1 mm or less. CT was used for segmentation of bones.